This is Honors Biology at Morris Catholic High School in Denville, New Jersey. The following tutorial will walk through a basic but extremely useful form of statistical analysis that will make the viewer the coolest kid in college. I'm your host and disembodied voice, Dr. Chris Charleglio, the chair of the Science Department, director of the Advanced Science Academy, and the former coolest kid in college. My mother said so. The laboratory experiment was titled Diffusion and Osmosis and can be found in the Morris Catholic AP Biology Lab Manual, page D&O 115. We begin by taking account of what data we collected in a lab long ago in a school far, far away. You will note that this was last used November 24th, 2020 by one of the students, and it has been fully filled in. The beautiful thing about the way this was designed is that it automatically takes the numbers that have been calculated as the basic statistical analysis that's necessary for a compilation that you find on this tab called Compiled. But let's just walk through it bit by bit. First of all, you had to have your group name. Second, you needed a data table title, which for some reason none of you did. Next was the label for which dialysis bag, because in Part B we were using dialysis bags, and which molar sucrose was in each dialysis bag. And that ranged from 0, the control, to 0.2 molar, 0.4 molar, 0.6, 0.8, and 1 molar sucrose in water. You took an initial mass in grams of each of the bags. Then you put them in water for a certain amount of time as dictated by the lab. Then you measured the final mass. It automatically calculated any change in mass using simple formulas. C4 minus minus B4. Then the change in mass in percent was calculated by looking at that change in mass versus the initial. These numbers are highly useful. You'll see that a really rudimentary and incorrect statistical analysis was done in this table. If you're interested in why this was the incorrect way of doing it, or why I might have put that on in the first place, you should come and see me, because it's an interesting conversation. I say jokingly that you'll be the coolest kid in college, but in all seriousness, if you know how to do this, you will be way ahead of the curve. I don't think that this is necessarily college level, that you are going to find it too hard to understand. I am walking you through this bit by bit so you can watch it over and over again and listen to what I say and see how to do it, because it's extremely useful. And if you know how to do that, you will be quite sought after in college by your peers. Anyway, so this is Part B, and you see this is the case for each group. So here's Group 1, here's Group 2, and I believe there were 4 groups. Yep, 4 groups. And then there was nothing in Group 5. Okay. For Part C, you took cores of a potato. We're not going to use temperature for this particular experiment, but it was set up very similarly, where you had what the actual independent variables were. Independent variable is simply what it's based on. 0, 0.2, 0.4, 0.6, 0.8, and 1 molar sucrose. And in case you're wondering, what does molar mean? Because we haven't had chemistry. Molar means how many moles, which is a form of measurement in chemistry, how many moles are actually in the solution. So a 1 molar solution is equal to 1 mole of table sugar, sucrose in this case. Differently from the way you did it in the bags, you actually put plain old fresh potato cores into these solutions in a cup. Okay, you took the initial mass first, and they were pretty consistent, 1.1 grams for most of the measurements that were made. And then because it was a weekend, I believe, I actually went and measured the final mass, and it automatically calculated the change in mass and what percent change there actually was there. Again, this is the incorrect way to do it. We are going to do it the correct way. So what this did was it automatically considered all of the independent variables, what sections did it, what part of the experiment it was, and most importantly, what the dependent variables were, what you actually measured. And it compiled it automatically into this very basic spreadsheet. Section. And that actually refers to how different sections of honors biology. This year, there's only one section of honors biology. That's why it all says ones. Then group, which group, 1, 2, 3, or 4, which concentration of sucrose was used for B or for C. And then these were the percent changes for group 1, group 2, group 3. You get the point. These are all of the numbers for B. These are all of the numbers for C. What we are going to do is we are going to take these data. Notice I said these data because data is plural. And we are going to move them into a statistical analysis program. The one that I would like you to practice on is called PSPP. Let me try that again. Is called PSPP, which is supposed to be a pun on the software I'm going to show it to you in, which is called SPSS. They work almost identically. So I am going to show you in the one that I've paid for, SPSS, and you are going to use if you wanted to do this yourself for practice, which you should, you're going to use PSPP. So here we go. Switching to the stats. On the right, you have over here the data set where it will go eventually. It looks like a spreadsheet, maybe with a lot more buttons. Over here is the output of what the analysis will look like. Okay, so I'm going to step you through this. The first thing you want to do is go to the tab on the bottom that says Variable View, and you're going to enter your variables. I think that since we only have one section, we're not going to use that variable. So the first one we're going to look at is group. Okay, that is going to be numerics fine because there's 1, 2, 3, and 4, but no decimal places. Say OK. And that's fine, that's fine, that's fine. Cool. And I'm actually going to make this a smidge bigger just for now, and then I'll shrink it back down again. There we go. All right, next one, 2, is going to be the concentration, so const sucrose. And I have to use a certain naming scheme for this because SPSS and PSPP don't like starting variables with numbers or characters. There's a way around this though. So that concentration should probably have one decimal place. And I'm just going to label this instead and say square bracket, not curly, square bracket, and say sucrose, and square bracket, and then put it in molar. So that's good. And that's a numeric value. Then your first measurement. So this is, let's say part B. Actually, why don't we say bags? Bags begin with a B. And then the next one will be potatoes. Potatoes. Or potato. Okay, and we'll, I think, hundredths of in decimals is perfectly fine. Let's do one significant figure. You'll learn about significant figures next year in chemistry. Okay, next thing I want to do is determine what type of measurement it is. Your group, there are 3 options. A nominal, meaning it's just a name and not a type of grouping that has anything to do with the data, or ordinal, which means that it is an independent variable, or scale, which means it's the dependent variable and the thing you measure. In this case, the group numbers were simply nominal. You were randomly assigned into a different group. But the concentration of sucrose was totally ordinal because those are the different types of sucrose it was put into. Since the bags and the potato measures are actual dependent variables, they are going to be scale. Now I could return to data view, and I can make this resume its previous size like that. Beautiful. And all I'm going to do is copy and paste from the Google sheet that we just had. There it is. Those are all of your data. Now you'll notice here's the concentration of sucrose, right? That looks right. Here is the percent change of the bags, and here is the percent change of the potatoes. So we're going to analyze this bit by bit. And the question then becomes, what exactly are we measuring? And so that's where a hypothesis comes in handy. What is the hypothesis about the bags filled with sucrose water? There are graduate students and postdoctoral fellows at major Ivy League universities who don't know how to do this basic thing. And here it is. The most basic hypothesis that you can come up with is, this is significantly different from that. So you might be like, well, what is he talking about? All you have to do is replace the words this and that with the things you're looking at, and you've got a functional hypothesis. It is not an if-then statement. That is not to say that if-then statements are useless. They're actually really, really useful in the results section to lead the reader through the paper. We thought that if this was true, then maybe this would be a result. That's not a hypothesis, but it is a great way to write. So you should keep those in mind when you write the results section of this paper. But when it comes to a hypothesis, you have to keep it simple, and it's looking for a difference. Okay, this is different from that. And you really specifically want to say, this is significantly different from that. When you say the word significantly, it means that you've run a statistical test and you are about as sure as you can be in science, right? We're never 100% sure about anything ever. We never know for 100% factual correctness. We just have lots and lots of evidence, and that's what this is doing. This is what you're practicing. So we want to know what the hypothesis is for, say, the bags. I think a simple one would be that bags are that the weight of the bags change significantly different. The change in the weight of bags, or mass of bags, is significantly different between different concentrations of sucrose. That was pretty clunky. So let's try that again. The change in mass between sucrose concentrations is significantly different. The change in mass between sucrose concentrations is significantly different. That is your hypothesis. You might want to make it a little more graceful than that in the paper, but that's the basic one. And for a statistical analysis, what you're testing is not the hypothesis, but the null hypothesis. The change in mass between sucrose concentration is not significantly different. It means that there is an absence of a result. That's what you are testing. Okay? So we're going to run the bags first. So we need to first look at what type of tests we might want to run. So we come back here, and you've seen this before. I've used this all through college, graduate school, my postdocs, and this actually comes from, I originally got it from the AP manual when I was your age. And it looks a little intimidating at first, but it's actually fairly easy to read. There are two main types of statistical analyses: comparative statistics and association statistics. Association means that you're trying to find a correlation between things, and we know that correlation is not causation, but it is informative. How you actually determine whether something is statistically correlated is using association statistics. We're not actually using those for this test. Remember, your hypothesis is that the change in mass between sucrose concentration is significantly different. You're comparing them. So comparative statistics would be a better way to do that. So then the next question is, are you looking at independent samples, or matched samples. And what that means is, are you looking at the same sample tested in each of the independent variables, or are you looking at different samples in each of the independent variables? So my friend Njideka Akinili from Swarthmore, she and I TA'd the Biology 2 together, and she had a great analogy. She was a very good runner, Division 3 cross country, I believe, and track. And now she's a fairly famous artist. You can look her up. But she used to say that the difference between them was like you wanted to know whether the cold weather would affect how fast you could run a, say, 400-meter sprint. So she used a track analogy. You want to know if temperature can affect how fast people run a 400-meter sprint. There's two ways to test that. You can take your whole team out, or a group of people from your class, and have them run the 400-meter dash in the middle of summer. Then you can wait a while and have that same group of people run it again. Okay, that means that they're matched. It's the same people, run it again, but this time in January when it's winter, and you actually compare the summer versus the winter. That's a comparative statistic using matched samples, but you'd have to wait a long time. A faster way of doing it, but less power, like this makes the most sense and is the most statistically powerful. But an easier way of doing it that is less powerful, but not invalid, it still works, is called an independent samples, where you are taking a track team here at Morris Catholic, having them run the 400-meter dash. Then you're calling your friend right now in January, or it's now February, and then call a friend who has a track team in Florida and ask them to run in the warmer weather. That would be independent samples. You understand it's less powerful because you're not looking for changes within individual people, but you are looking at the overall changes to the team. So in this case, since we are taking bags and we are only putting them in one of the solutions, this is an independent samples. So the next question is, how many groups do you have? Do we have 2, or do we have more than 2? Well, if we come back to here, we have 1, 2, 3, 4, 5, 6 different groups that were being tested. Just noticed a small mistake. It was missing this 2 here, so I put those data back. We're good now. So we know that there are 6 different groups. 6 different groups are more than 2. So the test that you're looking to run will be either an ANOVA, a Kruskal-Wallis, or a chi-square test. And so we need to then refer up to the top here to see what type of test those actually are. The first column is called a parametric test. They are arguably the most powerful test. They're the best at finding results and not giving you false negatives. Problem is, it does give you false positives. Non-parametric can be applied really to any data, but they're less powerful. And if you want more information on that, you should take statistics next year or the year after. The last category here is frequency tests, which are counts. We're actually going to use those a lot when we're looking at, like right now, we're looking at counting genes and recombination frequency. And this is actually a really good way to do it, to figure out if there's a significant difference. It's called a chi-square. It's the only test that you'll ever actually be asked to run on an AP exam for biology. That said, it's not the type of test that we would use on this because we're not counting things. Again, go back to the stats. We haven't actually counted anything. We're comparing the differences between stuff, not counting. So chi-square is out. That's not the appropriate test. So now you're down to ANOVA or Kruskal-Wallis. Now the kooky thing is that there are rules about running ANOVA, and the only way to know whether you can or not is to run the test in the statistical software. So in order to run a parametric test, you must follow two basic rules. The first rule is that the data must be normal. And I think we did this in class, but that means that the distribution must be normal. And I'm going to show you what to look for to figure out whether something is normal or not. The second criterion, the second rule, is that the data, the variances of data, so how variable the data are from group to group. So if I go back to my stat sheet, from 0s to 2s to 4s to 6s, you would expect their bell curves to be about the same. If they are significantly different, you have violated the second rule and you are not allowed to run the parametric test. So again, to run a parametric test, which is this column here, okay, anything in this column, you need to run have normal data. That's rule number one, and the data, the variances in the data cannot be significantly different from each other. They have to be about the same. Now, we've actually looked at those graphs before, what normal data look like versus skewed versus other types of characteristics. But the basic idea behind this is that parametric tests use a mean as the average, not the median, but the mean. And he uses standard deviation and standard error and confidence intervals. But he uses the mean. So if you remember, we drew at one point a graph where it was bimodal. It had a bump and then another bump. But if you took the mean of that, it was right in the middle where there weren't any cases, and it was not representative of the data, right? You wouldn't look at something that has two humps and then a data point right in between them and say, yeah, that's representative of the whole group. No, no, no. That's not how it works. Only if the data are normal can it represent the group. So how do we do this? Let's go to the stats, and now we're actually going to run some stuff. So the first thing that you want to do is you want to analyze, and you could do this on either window. You can come over here and do analyze also. We're going to do a statistics, descriptive statistics, and we can look at the frequencies of, we can do these both at the same time, but for clarity, I'm going to only do one at a time. We're going to do bags. Put that over as a variable. You do not want to display the frequency table. I know that, thank you. You don't want to do the frequency tables because they're very long and not really informative for you. What statistics would you like to run? Well, I guess I'd like to know the mean, the median, the mode. I don't think the sum is very useful. The standard deviation, maybe the variance, the standard error, if you're looking at a whole population. So the standard deviation is among the data. The standard error is when you extrapolate to a whole population. Because remember, the whole reason that we're doing this with samples is because I can't do it in the entire universe, right? There's a difference between a census and a statistical analysis. A census means that you are testing it everywhere at all times, everywhere. and that's impossible. You just can't do that. So instead, we use representative samples. So standard deviation refers to the samples. Standard error is an extrapolation to what it looks like in the population. The most important things here are the skewness and the kurtosis, because those tell you if the data are normal. So I'm going to click continue. Do I want any charts? Sure. Let's run histograms with a normal curve to see if the curve fits it. There's no format, style, or bootstrap. I'm just going to click OK. And so this is what you get. It gives you a table and tells you there are 24 pieces of data, which correspond over here to the 24 data points. It says that the mean for them is 32, so a 32% change in bag weight. And it's a positive number, so it's in the positive direction. It gains weight. And then standard error, medians, modes. these have various uses or usefulness. The last one, the one that we really care about here, is skewness and kurtosis. And in order for it to be normal, the skewness and the kurtosis must be between -2 and +2. So negative 0.271 is more than -2, and it's less than +2, so that works. Kurtosis is -9. I don't know why that pop-up keeps coming up. -0.986, which again is more than -2, less than 2. So that works also. And here's what all the data look like with a normal curve. So it looks like these data are normal, right? No. We didn't do it right. And I always do this on purpose. I did it wrong on purpose. Who cares what all the data look like all at once? There's no way to tell whether things are different if you're looking at all the data all at once. So what you need to be doing is looking for normalcy or normality within each group. So in order to do that, so if I'm just going to minimize that since it's useless. Keep it, but I'm going to actually rename it. Let's call it useless. Useless. There we go. Okay. So instead, first thing I want to do it correctly is I want to split the file. And I want to split it I want to split this either by comparing the groups or by organizing the output by groups. It gives you the same data either way. It's just a different way of presenting it. You want to use the groups that they're actually split into, which is the sucrose concentration. So I'm going to click compare groups based on sucrose and bring that over. And so I'm going to split the data by that. So it's going to say it's going to sort cases and split them by their sucrose concentration. Now, if I go and I run the same statistical analysis I just did, it's going to, I'm going to show you, I'm going to do it, then I'm going to delete it because it's going to be wrong, and you'll see why in a moment. So I'm going to, it keeps all the same settings we had last time. See, all the same things are connected. You know, it might be worth putting quartiles. Yeah, let's put the quartiles. Continue. We'll leave the charts for now, but that's going to be the mistake. It'll be better to do none, and you'll see why in a moment. And that's about it. So I'm going to say OK. All right. So what this did was it broke down by all of the different types. So 0, 0.2, 0.4, 0.6, 0.8, and 1. And then it gives you histograms, and those histograms really aren't that useful, which is why I was going to turn those off. Now, just to give you an idea, I'm going to actually select all these and delete all those just to make it clearer. What would have happened? See how it's presented like this, where 0.0, 0.2. I'm going to show you the other way of doing it, and maybe you'll like that better. So I'm going to get rid of that. Okay, so here we go. I'm going to minimize that for a moment. I'm going to go to Data. I'm going to split the file. We're still going to use this, but instead we're going to organize the output by groups. Again, it's going to be the same analysis. It's just going to be presented different to you. I'm going to say Analyze, Descriptive Frequencies. This is all selected correctly as you saw, so I'm just going to hit OK. Oh, I forgot to, oh, no, I forgot to, hold on, I'm going to do it again. But this time I'm going to turn off the charts since they're not that informative. Frequencies. Charts, I'm going to turn them off. None. Thank you. Okay. So this is the way it did it when you organize by groups. So it looks almost the same. Maybe this might be clearer. So again, here's the old version where they put them all in the same table. It's all the same data, right? So it'd be 0, the control, the negative control, 0.2, 0.4, 0.6, 0.8, and 1, and then the same data again analyzed and just displayed differently. So you can choose whichever one you like better. I'm going to, I don't know, this one seems clearer, so let's use that one. I'm going to delete the old one. Delete that, delete the log. There we go. Okay, so what this allows me to do is look by group. So in this first group, which is the negative control, 0.0, or 0, just 0, it's going to tell me the skewness and the kurtosis. And right off the bat, you see that the control violates the rule. Negative 5.9 is less than negative 2. Right away, you know it violated the rule. You cannot run parametric statistics. You cannot go over here, look at this, and run an ANOVA, because in order to do that, you had to meet the rules. You had to meet the criteria and not violate the rules. But you did. We did violate the rules. Going back to the stats, it is negative 5.9. Now, just for the sake of argument, let's look and see what else. Skewness is good. Kurtosis was bad. All right, what about 0.2? Skewness good, kurtosis bad. So that violated it again. What about 0.4? Skewness is good, kurtosis is good for 0.4. 0.6, skewness is good, kurtosis is good. 0.8, skewness is good, kurtosis is good. And finally, the last one, skewness is good, kurtosis does violate it, but not by much. So it doesn't really matter. You violated the first rule of parametric tests, so you can't run it. Instead, you have to run a non-parametric test. But there's a weird thing. There's a weird thing in this program, and I think PSPP does the same thing, where in order to get the data analyzed correctly, you actually have to run the ANOVA, even though it violates the rule, just to get one of the outputs it does. So I'm going to show you what that looks like. So now, since we're going to be comparing the 0 versus 0.2 versus 0.4 versus 0.6 versus 0.8 versus 1, I need them all to be back in the same data set and not split like we just did. Remember, when we just did this, we actually split them out so we can just get descriptive statistics about them. Now we don't need that, okay? And now the mean is no longer useful, by the way. So what we're going to do is we're going to put them back together. I'm going to say data. I'm going to split the file and unsplit it. Analyze all cases. Say OK. All right, so it says split file off. Now it's good to run the actual data analysis. So the first one that I'm going to run is the one that I'm not allowed to use. I'm allowed to run it, but I'm not allowed to use it, and that's an ANOVA. So what I go to is compare means. It's called a one-way ANOVA. It's going to ask me, what is the factor that I'm using? The factor is the sucrose concentrations. And right now, we're going to run just the bags. Now, the thing is that you could run actually both of these at the same time and really save yourself a lot of heartache and time having to analyze these. But this is the first time that you're ever doing this. So I'm trying to do this slowly, methodically, and make sure I explain every little bit, right? The dependent variable is the weight of the bags. The factor by which you are splitting them is the concentration of sucrose. Skip the contrast, go to post hoc. Here's what matters. Because we already know that this is an abnormal data set, we cannot compare 0 to 0.2, 0 to 0.4, 0 to 0.6, 0 to 0.8, 0 to 1, and then 0.2 to 0.4, and then 0.2 to 0.6. Like all these little pairwise comparisons, you can't do them individually. It has to be done in what we call a post hoc test. Post hoc means after you've done the omnibus test, afterwards. That's what post means, right? After. So this is a multiple comparison, but you already know that you broke the rules, so you can't use any of these. You have to use the ones where you don't assume equal variances. So I prefer to use Dunnett's. That's what we're going to use. And notice that the significance level is 0.05. We've talked about this with p-values. All right, p-values are have to be less than 0.05 in order to be considered statistically significant. So I'm going to just do this. I'm going to say continue. Let's look at the options. Yeah, I like the descriptives, but the whole reason you're running this, besides that Dunnett T3 that I just said, this is the most important thing. Homogeneity of variances. That is what we call the Levine's test. The Levine's test is also checking the second rule of a parametric test, right? You violated the one about them being normal. They're not normal data. But what about their variances? Does that violate it anyway? So I'm just going to show you, let's say that you had met all of the requirements for normality or normalcy, and you met all the requirements, and now, so rule 1 was good, which it wasn't, but let's pretend it was. The second thing you'd have to look at is this, homogeneity of variances. So I'm going to hit continue. I'll explain that again when I hit OK. Here we go. All right, so this gives us some descriptive statistics just like you got above, and they should all be the same statistics. So like if I look at the, I don't know, let's look at the mean value for 0. The mean is 11.193. So if I go over here, I open that up, and I say 0.0, what's the mean? 11.193. Yes, it's the same data, and that's a good check to make sure you didn't screw anything up. Okay, so we can't use this. This is the actual statistic, but remember you violated it, so I'm going to delete it. Haha. So this is the Levine's test, Levine's statistic for homogeneity of variances. It's asking, does the variance of one of the groups, is it different from any of the other groups? And in order to answer that question, you would have to look at the significance. If the significance is less than 0.05, which all of them are, they are significantly different. So if you were like concerned that, oh, I really wanted to run an ANOVA parametric test, well, now you can feel a little better because you actually violated both rules. And don't think that you did anything bad here. This is just the data are what the data are. And in this case, they violated the rules, therefore using a parametric test wouldn't actually tell you anything. It would lie to you, really. It would actually tell you false information. So you don't want to run tests for which they're not prescribed. So that's interesting. The homogeneity of variances are violated, so that's pretty useless now. So what I'm going to do is I'm actually going to delete the descriptives because we don't need them. We already have them. I'm also going to delete this homogeneity of variances because you violated it and you're not allowed to use that. What matters here is this post hoc test. And this is you only use this if the main non-parametric test is significant, and we haven't run that yet. So let's do that. Let's run that test for realsies. So I'm going to go to Analyze, Non-Parametric Tests. Hold on, I need to switch back to here. Okay, I'm going to say Analyze, Non-Parametric Tests. I'm going to run a legacy type, it's called. I'm going to go to, this is hard to see, so what I'm going to do is I'm actually going to drag this to take up the whole window. There we go. So again, Analyze, Non-Parametric Tests, Legacy, and we are going to run, we know it's independent samples, and it's more than 2, right? So 2 independent samples, it's more than that. So we just say K independent samples. Cool. What's the test variable? It's going to be the weight of the bags. What's the grouping variable? Sucrose. And it asks you to define the range. The minimum is going to be 0, and the maximum is going to be 1 molar, right? So 1. Continue. It says we can run a Kruskal-Wallis. Remember, over on the table here, this was the test. Kruskal-Wallis Test. So that's what we're going to use. Okay. So Kruskal-Wallis Test. And I believe we would like the descriptives and the quartiles. That'll be good. I'm going to skip this exact test and just hit OK. And there it is. This is the most important thing we've run so far. Going back to our hypothesis, the change in mass between sucrose concentration is significantly different, is what was tested. So what you do is you look down here at test statistics, and it says this was the H value, 9.6. This is the, that double click is really annoying. This is the 1 is the degrees of freedom, okay, and the asymptotic significance, or this, the p value is 0.002. Is that less than 0.05? Yes, it is. That means that your bags of sucrose between concentrations is significantly different. That's called the omnibus test. And that's That's the most important result. This better end up in the paper. It's the most important result you have. So if you were presenting this to me, you could actually say, yes, the bags, the weight of the bags, the mass of the bags was significantly different after the experiment, and they were significantly different from each other depending on the concentration. You can say that now because it says it right here. P is less than 0.05. Okay. Anadditionally, if you want to be more specific, you have the Done-It test up here, the post hoc test that we ran with the ANOVA. And so what this does is it compares each concentration. Here's the negative 0 versus 0.2, 0 versus 0.4, 0 versus 0.6, 0 versus 0.8, 0 versus 1. And it does this for each one of the concentrations, bit by bit. All you have to do, the quick way of doing it, is looking for the little stars. Anytime there's an asterisk, it means that its p-value, whether or not it's different from each other, its p-value is less than 0.05. How do you know? Well, you can look over here where it says significance, which is the p-value, and it says, for like this one, 0 versus 1 is 0.011. Is that less than 0.05? It is. Therefore, you can reject the null hypothesis that they're not significantly different, and you can say that 0 and 1 are, in fact, significantly different. So think back to that middle schooler who's going, they're different. Well, I want to know how you know that they're different. Now you can say, having run this statistical test, that they are indeed different. It was a non-parametric statistical test. Okay. Next thing is the potatoes. So what I'm going to do is I'm going to do the whole process quicker. Okay, and then I'm going to give you these PDF files and this video, of course. And I was hoping to do this video in less than an hour, and right now we're 42 minutes. So I think we're in good shape. So I'm going to do everything all at once. And you'll have to watch and pick it apart. So again, we're looking at these data here, but instead of bags, we're looking at just potatoes. Okay, so I'm going to put that back. So first thing I want to do is split the data again so that I can get my descriptive statistics. Split file. But this time we're going to organize output not by that, and it's going to be right. Yeah, that looks right. Say OK. So it's now said split the file. Cool. Analyze, descriptive statistics, frequencies, but this time we're not going to do bags. We're going to do potato. Beautiful. Okay. All right, so what we're looking for is violation of rule number 1. It has to be greater than -2 or less than +2. So skewness is good, kurtosis is good. Skewness is good, kurtosis is good. Skewness good, kurtosis is it's over. I'm going to make an executive decision right now and say, well, that's acceptable for now. Yeah, no, that's good. Okay, so then skewness is good, kurtosis is good. What about 8? Skewness is good. Kurtosis is violation. Boo. Okay. And then over here, skewness is good. Kurtosis breaks it again. Okay, so you can feel pretty good that, once again, parametric tests, while you would rather run them because they're more powerful, right? While you would rather run a parametric ANOVA because it's more powerful, you have to follow the rules. And the rules are that the data must be normal. That's rule number 1. And rule number 2 is they can't that the variances can't be significantly different. So I know right off the bat that I'm going to have to run a non-parametric test. So I'm going to do that. So I'm going to say data and split file. I'm going to unsplit the file and analyze all cases. So say OK. So this time I'm actually going to just run the non-parametric test first. Analyze non-parametric. Non-parametric, legacy dialogues, k independent samples, and this time we are going to run the potato with the same concentration as sucrose, and it's going to be a Kruskal-Wallis. The options are already selected for us, so we're going to click OK. And just like the last one, you can see that this is actually significant? Is it less than 0.05? Yes. And what that means is those potatoes are actually the different concentrations of sucrose make the potatoes significantly different from each other, and that's pretty cool. You can now say that, like instead of being a middle schooler who doesn't know what they're talking about and is just saying, yeah, they're different. You can actually now say, yeah, they actually are different. I statistically analyzed it. The next thing to do, though, of course, is to you want to know is like which concentration is different from which other concentration, which this doesn't tell you. This just tells you there's a difference, which is great. You need that. You can't proceed without that. But now I actually have to run. This is weird. I have to actually run the ANOVA, and the danger of this is that it shows you the result of the ANOVA, and so I have post hoc again done at T3. And so I learned statistics from Dr. Karen Gamble, who's a professor now so this was while we were at Vanderbilt. She was a postdoc when I was a graduate student. And she was adamant about this. And she went on to be a professor at the University of Alabama, Birmingham. But she used to say, okay, you can run this in order to get the post hoc test and the Levine's test, but don't you dare look at the ANOVA results. So if I click OK, like the descriptives, those are innocuous, but we already have them. That's fine. I'm just going to delete them. And this is what we did. We wanted the Levine's test, the homogeneity. And it's this thing that was the danger. See how it says like 0, 0, 0? No, you shouldn't see it because you're not allowed to look at it. Stop looking at it. I'm deleting it. See, she'd be really like aggressive about that. You're not supposed to look at it. You violated the rules, so don't think that it tells you anything. It doesn't. It does not tell you anything. But this is interesting, actually. So the Levine's test is saying for the potatoes that they're not significantly different from each other. The variances, the bell curves, the shape of the normal distribution for your little sample at each concentration of sucrose is not different from each other. This is just I don't think this is something you could say in the paper, but from my experience, that means you guys did a really good job of cutting the potatoes and then putting them in the right amount of solution at the right temperature and sealing it. And like, there's not a lot of, again, this is my interpretation, there's not a lot of human error there. If their variances are essentially the same, that's pretty good. So good job. But really what's interesting, after you find out, okay, I've got the omnibus test, right? The thing that includes all the data, and it says that there is a significant difference, but it's not specific. Well, here in the Dunits, you actually get the specifics. You can see 0 versus 0.2, 0 versus 0.4, 0 versus 0.6, et cetera, et cetera. And the quick way of looking at to see if they're different is you just look for the asterisk. And it looks like everything is different from everything else except for 0.4 to 0.6 is not different. 0.6 to 0.8 is not different. And 0.8 to 1 is not different. But other than that, everything's different from everything else. And again, a quick way of looking at that is the significance level here if it's less than 0.05. So that is pretty impressive. I think that I think that you're ready to go. And so this is step number 1 for analysis. This is for the first paper you're going to write. And I think that it's really important when it comes to writing the paper for you to read chapter 4 and chapters 4 and 9 from Jan Pachenik's book, which I have provided to you in the lab online. And I am going to provide you these in a PDF that's going to be if you haven't watched this video, it's going to be very intimidating for you. You'll be like, what does all this mean? Well, if you watch the video, and I'll say this in class, of course, but if you watch this video and listen to me and assuming that it actually works, you will be able to understand every one of these steps. I think I might do a combo where I do both of them at the same time, just so you can have those data that are a little more compact. Since I have time, I still have 11 minutes before my 1 hour deadline. So I'm gonna actually do that. I'm gonna do all of them together this time. So I'm gonna say data, split file. I'm going to organize by groups. Say OK. I'm going to analyze. I'm going to go do that. I'm going to go frequencies, but this time I'm going to use bags and potatoes, but I'm going to do them in the correct order. So I'm going to select both of them, put them over here. Excellent. Say OK. There they are. And you can see now they're side by side. So you can actually look at the 0.0 for bags and potatoes. Bags and potatoes all at once, which I think is, for me, that I'm experienced, it's a lot easier for me. The next thing is you're going to go data, split file, and you're going to analyze all cases. You're going to unsplit the file so you can do an actual comparison. And we already know that you are supposed to run a non-parametric test, legacy dialogues, k independent samples. This time we're going to run both of them at the same time. Beautiful. Say OK. And there it is. It runs both of them, says both of them are significant. Then I am going to say, okay, analyze, compare means, Way Anova, and again, I'm going to run both of them at the same time. So you see, this is, so the other, walking through this one bit by bit can be a little tedious, but the beauty of a graphic user interface like this is that you can actually run all your tests at once. And so when you're a graduate student, you're on this, unless you know how to code in R, which is way better. You should try to learn that. It's way better than SPSS. My brother, who is a biostatistician, he knows R. And whenever he sees that I'm working on SPSS, he looks down his nose at me and is like, oh, you use that? He's a professor at George Washington University. And he's right, because this program sucks compared to R, but you can, if I can convince you to learn R. So here we go. Here's our descriptives. Here's our homogeneity. We're going to delete this because that didn't matter. And then here are, split between bags and potatoes, the post hoc test. And we're done. And that's it. So all you guys have to do is write a paper now. Oh, I did that in a minute and a half. Wow. So that's it. I hope you've enjoyed this presentation of diffusion and osmosis statistical analysis in the honors biology class at Morris Catholic High School. I'm your host, Dr. Char. Over and out.